Assalamualaikum warahmatullahi wabarakatuh to our both panel, Dr. Muhammad Akmal Hashim and Dr. Karunisa Ahmad Kamil. So our group will present about identification of fish population in Kuala Perlis.
Introduction. Aquatic environments are vital for biodiversity, ecological balance, and livelihoods. Malaysia, a global biodiversity hotspot, hosts diverse freshwater and marine fish species. Yet, areas like Kuala Perlis remain under research. Located on peninsular Malaysia northwest coast, Kuala Perlis estuarine and coastal ecosystems support fish populations crucial for local fisheries. However, challenges like overfishing, pollution, and habitat degradation threaten this ecosystem. Research from other Malaysian regions highlights significant species richness, such as 186 fish species recorded in Perak. Understanding local fish diversity and environmental influences is essential for conservation planning. This study aims to catalogue fish species in Kuala Perlis, assess their distribution, and provide recommendations for sustainable management and conservation efforts. For literature review, Kuala Perlis, located in northern Peninsula Malaysia, is home to a diverse range of fish species supported by its coastal waters, estuaries, and mangroves. These habitats provide critical resources for both commercially valuable and ecologically significant species such as barramundi, mullets, and gobbles. However, the distribution of these species is shaped by factors like tidal movements, salinity, and human activities such as overfishing and pollution. Efforts to conserve fish biodiversity in Kuala Perlis focus on protecting mangrove and establishing marine protected areas, which have shown positive results in increasing fish population. Sustainable fishing practices are also crucial for ensuring long-term ecological balance and supporting local livelihoods. Research continues to play an important role in understanding and addressing the challenges facing fish diversity and distribution in this region.
Methodology. For the sample collection, it starts with the field selection, where we choose Kuala Perlis as our primary site for this research due to its rich biodiversity and the environmental factors that influence the fish species in this area. This setting provided an ideal foundations for the observations and sample collection. The second one is fish capturing. Fish were captured directly at the sea of the Kuala Perlis using standardized sampling techniques such as gill net, which trap fish by their gills, and scenes by the fishermen, which encircle and capture shoals of fish alive. This method ensured the sample reflected the diversity of marine ecosystems. So, for the next step is the market observation. The local fish market near the sea was observed for the use of fishing technique and species that are commonly caught. Fishermen were observed using traditional and modern methods such as gill net and deep net to supply both and local and international market. Other than that, by comparing the fish species commonly sold at this market, the data that was collected can identify which species has a lot of abundance in this area. And the last one is sample selection. Finally, selected fish specimens were chosen based on their diversity and relevance to the study. These were carefully preserved and brought back for the further laboratory analysis to identify species and evaluate biological characteristics. Analysis, we start with the fish preservation. The fish collected during the field survey were preserved in a laboratory refrigerator under controlled conditions. This step was essential to maintain the quality of the sample and to facilitate accurate observation. And then, we undergo a freezing process. The samples were refrigerated for a week to freeze them, making them firmer and less fragile. This process prevents damage or degradation and ensuring the fish remain in excellent condition for the detailed analysis. So, for the third one is the observations of taxonomic characteristics. Key taxonomic features of the fish, such as body shape, color patterns, gill structure, and fin morphology were examined. This trait helped accurately classify the fish into their genus and species using taxonomic keys. For the next one is the evaluations of biological characteristics. The analysis extended beyond physical traits to explore how each species fit into the local ecosystem. Data on habitat preference and feeding behaviors provided insight into the ecological roles in maintaining biodiversity in Kuala Perlis. And the last one is ecological analysis. Finally, we analyze the environmental factors like habitat structure, water quality, and food availability. This allows us to understand the relationship between these variables and fish diversity, and aiding in biodiversity management and conservation strategies.
So our first identification of fish is sharp mackerel, or also known as combo. The scientific name of sharp mackerels are Scomber japonicus, and it is a family of Scombridae, and the genus of sharp mackerels is Rastrelliger. And the second species is mackerel scud, or also known as selayang. The scientific name of mackerel scud is Decapterus mackerellus, and the family of mackerel scud is Carangidae, and the genus is Decapterus. Both of the species is the phylum of Chordata. Our third identified fish is torpedo scud or hard-tailed scud, also known as chencharu. The scientific of torpedo scud is Megalapis cortilla, and it is in the family of Carangidae and the genus of Megalapis. And the fourth one is Indian mackerel, or also known as borat. The scientific name of Indian mackerel is Rastriliga canagurta, and it is the family of Scombridae and the genus of Rastriliga. And both of these fish is also under the phylum of Chordata. And our last identified fish is Spanish mackerels, or also known as tengiri. The scientific name of Spanish mackerel is called Scomberomorini. The family of Spanish mackerels is called Scombridae, and the genus of Spanish mackerel is Scomberomorus, and it is under phylum of Chordata.
So for discussion, this study aimed to determine fish species and diversity, and also to evaluate environmental conditions that influence fish species in Perlis. By doing some sampling method, which are the service and laboratory analysis, four types of species were recorded. Next is factors of environmental conditions that influence our fish species in Perlis. First is water temperature. Water temperature can affect metabolic rates, reproductive cycles, and migrations behaviors. Second, dissolved oxygen. Low levels can cause physiological stress and fish mortality due to interference or organic decay. For ecological use of these fish in local ecosystem, first is rod and food wheel, which is maintain biodiversity and ecosystem stability. Second, migratory fish, which is transfer nutrients between ecosystems. And lastly, overall contribution is to enhance productivity and stability of habitat.
And now we have reached the conclusion part. This study identified and cataloged fish species in Kuala Perlis while exploring how environmental factors like water quality, temperature, habitat, and food availability affect their distribution. Stable populations of key species such as Spanish mackerels highlight the area's ecological and economic value. Field surveys and market observation emphasize the need for sustainable practices and conservation efforts involving the local community, and implementing effective management can help protect the region's biodiversity and maintain its ecological balance. This research provides a strong base for future studies and conservation initiatives to ensure the health of the aquatic environment and support local livelihoods.